Hey, Mr. P here. In this video, we're going to talk about how traits are inherited, specifically heredity, and we're going to start this entire kind of heredity series talking about Mendel and why he was so instrumental in the development of genetics as we know it, and how he ultimately became the father of genetics. So, what did he do up front to lay the foundation for our total understanding of genetics and our understanding of heredity, and specifically how are traits inherited from Mendel's time? So let's get started. So we are starting this discussion with a quick introduction of Gregor Mendel and why pea plants were used in order to study human genetics. Obviously, this class is a human genetics or a human heredity course, and so it may seem kind of counterproductive to look at not only Mendel but peas, because it's obviously human genetics and peas are not humans. So why are we starting with Gregor Mendel and pea plants? And we have to understand his experimentation and how he organized his experimentation, organized his trials, organized his experiments in order to uncover the fundamental principles of genetics. You cannot understand or cannot begin to understand human heredity without understanding the fundamental principles of genetics. And he became infamous and really paved the way for our understanding of human genetics by utilizing pea plants for test subjects in the experimentation that ultimately gave way to the fundamental understanding of heredity as a whole. Humans cannot be used as test subjects. We can utilize a variety of different experimental organisms like mice, chimpanzees to an extent, bacteria, plants, of course, but we cannot use humans in experimental genetics because of the ethics involved with human experimentation, and humans aren't good test subjects anyway. So even if we could and did get permission to utilize humans in genetic experimental trials, humans don't make very good test subjects because, one, humans don't have very many offspring, right? Humans may have up to 3 offspring per reproduction cycle. Most of the time humans have a single child per reproductive cycle. But pea plants have hundreds or thousands of offspring per reproductive cycle. And humans take way longer to sexually mature. And so it would take tens of years, if not 100 years, to get multiple generations of humans and to look at multiple generations of traits that are expressed by those humans. Whereas because pea plants take days, we can get really good valid data over a lot of different offspring relatively quickly, within months, if not within a year or two, depending on how many trials or how many generations you're taking your experiments to. But pea plants make much better test subjects based on the number of offspring they produce, as well as the generation time and the turnaround time we have in those plants versus humans. We must start with an experimental system in which the mechanisms of inheritance are clearly defined, which he did. Okay, he was very methodical on making sure his original parent strains or his original parent plants were pure breeding or true breeding for their particular trait. He then always self-crossed the F1s regardless of the traits that were expressed, and he was methodical about keeping generations organized by P, F1, F2, F3, F4, F5, and he was really good at calculating probabilities and organizing his data in a way to analyze the fractions and the probabilities of the traits that were expressed. Okay, you have to have a clearly defined mechanism of inheritance up front so that you can attribute any of the inheritance or any of the deviation of traits or the variation of traits to the cross that you are attempting to breed. So a couple questions surfaced. Does each parent contribute equally to the traits of the offspring? Was one of the big questions that he was trying to discuss, okay, or discover. In plants and most animals, the female gametes, the egg cell, are always much larger than those of the male cell. So, at the time of Mendel, it was logical and widely debated question about whether or not females and males contributed equally. If you look at an egg cell for a human, for instance, it is substantially bigger than the sperm cell. And so it, like I said, was very easy to assume that because the egg cell may be 1,000 times bigger than the sperm cell, maybe the female contributes 1,000 times more, or 100 times more, or even at least double from that of the sperm cell. Okay, do they contribute equally? Obviously, now we know they do. Okay, it doesn't matter about the size. The egg cell can be huge, and is. The sperm cell can be small, and is. But they do contribute equally. Now, they contribute equally in terms of the genes. They contribute equally in terms of the genetics. They contribute equally in terms of the DNA. They don't, however, contribute equally of the organelles or the cytoplasm, because obviously the egg cell is in fact bigger, and so it therefore contributes unequally in terms of the cytoplasm and the enzymes and the organelles and the cytoskeleton and the infrastructure that the cell, the developing zygote, will have. Obviously all comes from the egg, but what the sperm does bring to the egg is an equal contribution in terms of the DNA that the sperm cell is carrying. Okay, do traits in the offspring result from blending of parental traits, or are they inherited as discrete units? There are situations in which some traits are blended. Okay, we call those non-Mendelian traits or non-Mendelian mode of inheritance. But when we talk about Mendelian genetics or Mendelian inheritance, the traits that are expressed, that are coded for by genes, are inherited equally and are inherited as discrete units that we call genes. You can have multiple different forms of the gene. We call those alleles, and we're going to get to some common language and some common vocabulary in a little bit. But we know that most of the genes that we inherit from our parents, mom and dad, are going to be inherited equally from mom and dad and are going to be inherited as discrete units that we call genes. Okay? That's a little bit about Mendel. That's a little bit about his start, the importance of our understanding of Mendelian inheritance. Obviously, we didn't get into any in-depth scientific processing. We didn't get into any in-depth crossing. That's all going to come in later videos. I just wanted to give a general overview of Mendel and why we have to start with Mendel and the importance of Mendel as we talk about, or as we begin to talk about, Mendelian inheritance, so that we understand the foundation of inheritance that gives rise to our understanding of human heredity. That's all we have. Bring your questions to class. See ya.